For my local expert project, I will be discussing Ion Torrent. What is Ion Torrent? Ion Torrent is a form of next-generation sequencing that utilizes DNA sequencing based on the detection of hydrogen ions that are released during the polymerization of DNA. It is also a form of synthesis by sequencing. The sequencing of DNA is done using a semiconductor chip. How does Ion Torrent work? The process begins with a target DNA molecule that is being sequenced getting broken up into approximately 400 base pair single-stranded DNA fragments and two different adapters alongside biotin, which are then added to the ends. Next, the resulting single-stranded DNA library is individually attached to a magnetic bead via the streptobiotin and biotin interaction, which is then amplified via emulsion polymerase chain reaction. As a result, millions of the same single-stranded DNA fragments are bound to a single bead. Afterwards, each bead is placed into a pH-sensitive chip containing thousands of wells. In this figure, we see Ion Torrent sequencing DNA library preparation and process. In figure A, the fragmentation and denaturing of the DNA and the addition of two different adapters and biotin. In B, we see the binding between biotin on the single-stranded DNA and the streptobiotin on the magnetic bead. In C, we see the emulsion PCR to result in a magnetic bead coated with millions of copies of the same single-stranded DNA. The semiconductor chip has millions of wells covering pixels that capture chemical information, DNA sequencing, and translate it into digital information. The sequencing process starts when the sample of DNA is cut into millions of fragments. Each fragment then is attached to beads. The beads are then deposited into wells of the chip. The chip is then flooded with one of the 4 DNA nucleotides. Then a nucleotide is incorporated into a strand of DNA. A hydrogen ion is released in the same process. The release of the hydrogen ion changes the pH solution in the well. An ion layer measures that change in pH and converts it to voltage. The voltage change is recorded, and that indicates the nucleotide was incorporated and a base was called. The process is then repeated with different nucleotides washing over the chip. This process occurs simultaneously across the wells. So to begin sequencing, one of the four DNTPs is added and washed away, depending on the signal and depending on if a signal is detected. Each time a DNTP is successfully incorporated onto the elongated strand by DNAP, hydrogen ions are released, resulting in a decrease in pH that is detected in the pH-sensitive chip. pH doubles, and this indicates consecutive DNTPs being incorporated. Lastly, the DNA is analyzed by a computer in order to obtain the DNA sequence of the original DNA molecule. Ion Torrent sequencing. So in A, we see the addition of one of the 4 DNTPs to the reaction sample results in the incorporation of one glutamine into the elongated strand and then the release of one proton. In B, we see the addition of one of the 4 DNTPs results in the incorporation of 2 thymines into the elongated strand and the release of 2 protons. And then again in C, we detect the detection of changes in pH that provides information on the sequence of the target DNA. How does Ion Torrent compare to what we learned in class? Ion Torrent uses synthesis by sequencing. This is similar to what the Illumina platform uses. As well as Ion Torrent requires library preparation with adapters. Again, we learned this with the Illumina sequencing. Unlike the Illumina sequencing, Ion Torrent uses pH versus fluorescence. And unlike the Illumina sequencing, the reads range from 200 to 400 base pairs compared to 150 base pair reads with the Illumina sequencing. Disadvantages of Ion Torrent. One disadvantage is the need to use control kit from Thermo Fisher. This limits you on the ability to use other control kits because the platform requires this type of kit to work. Another limitation of Ion Torrent is the presence of homopolymers, so repeats of the same nucleotide. If there are over 10 of the same bases in a row on the template DNA strand, the platform can no longer make accurate calls. Also, the approach is dependent on emulsion PCR to amplify the DNA plates, which can be lengthy and may introduce biases. Ion Torrent advantages. Ion Torrent sequencing became the first DNA sequencing technology to completely eliminate the need for fluorescently labeled DNA and imaging devices, making it faster and cheaper than other alternatives in the market at the time. In addition, the fragment length increased from 50 to 400 base pairs, contributing to an even faster sequencing speed. Besides offering fast readout times, Ion Torrent's sequencing is also highly accurate. It has a 99.6% accuracy rate. Faster readout times compared to the Illumina sequencing, Ion Torrent runs in 2 hours versus the 27 hours on the Illumina. Additionally, the technology managed to be relatively cost efficient in comparison to other options available in the market. When looking at the Illumina mini-seq, it costs $128,000 compared to the Ion Torrent, which costs $80,000. Ion Torrent controls. Like I mentioned in a previous slide, Ion Torrent uses a control kit from Thermo Fisher. These kits contain controlled genomic DNA and control libraries of 200 and 400 base pair run configurations using an Ion PGM semiconductor-based sequencing platform. The kit provides for greater experimental control through the use of quality control reagents for the semiconductor-based sequencing workflow, and allows you to assess sample quality, save time and reagents, and maximize productivity. The Ion Shot system can be used to automate the emulsion PCR process, removing the need for lengthy and multiple-step preparation procedures with water and oil emulsification. Now on to some data. Here, this paper compares Ion Torrent versus a HiSeq 2000. Here we see the total reads number and the total basis number between the both groups. In the max read length, we see that Ion Torrent has a max read length of 201 compared to the HiSeq at 90. The min read length is 15 on an Ion Torrent compared to a HiSeq at 90. The map read number is comparable between the two groups, but the map rate percentage is higher on the Ion Torrent at 95% versus the 76.57% of the HiSeq. Again, the Ion Torrent has a higher coverage rate at 96.5% versus the 93.11%. In this paper, we see the flow signals and their position on the SAM-BAM file. The upper part shows an example DNA fragment to be sequenced by an Ion Torrent machine. The key and barcode sequence are ligated to the fragment. The key sequence, TCAG, is a control sequence to ensure correct sequencing. A barcode sequence is added to a certain group for fragments. The use of barcodes makes it possible to sequence the DNA of different samples/patients in one run. The low part of the figure shows a systemic representation of the fields in the SAM file. The SAM file is a non-binary readable version of the BAM. The header part includes the flow cell and the key sequence. Each line in the SAM file represents one read aligned to the reference genome. The remaining rows include the read information in the tab separate format. We show only the columns/fields of relevance in this paper. The fields include the read ID, the physical position, and the CGAR string, which represents the alignment, the basis of the DNA sequence in the read the quality field, and the flow signals in the CM file. Lastly, in this figure, we see a readout from an Ion Torrent. So this figure is maintaining sequencing performance in the presence of scaling. The figure shows a typical 200 base AQ20 90% aligned accuracy read from a Proton 1 run. At left are normalized incorporation signals, and at right are the associated base calls aligned against the reference genome, so lower sequence tag. The 203 base sequence read contains two sequencing errors indicated by misalignment at base 97 and base 112. This has been my local expert project on Ion Torrent. Thank you.